Hello everyone. Today we will be talking about protein purification, and I will try to explain it in a very simple way, the way that I understand it at least. I do not come from a biology field or anything like that, so hopefully this would help for someone too. So we can think of this four-step system of basically how we practically break down protein for purification. So the first step is called cell and tissue disruption, okay? So this is when we first take like our full cell. So imagine we have here our cell. Let me just get a cell real quick. So imagine we have here this cell, and as you can see, this cell has like a lot of stuff inside, right? So basically what we want to do is we want to take like some specific things from here, okay? So let's say we want the nuclei and some proteins and all of that, and some other organelle of the cell, but then we want to discard everything else. So this is basically our end goal of this entire thing. We try to extract organelle and protein from cells and tissues without basically mixing the other stuff too. The way that we do disruption is actually you can think of as threefold, okay? So it depends on the type of disruption, and we have here three distinct types. It's very simple. We have here basically gentle, harsh, and lastly also cryo grinding. Now, actually, this depends on the cell that we're trying to, get this from. So, generally speaking, harsh cells can be something like animal tissue or anything like that. And then cryo grinding is for very, very difficult cells. So, this could be mainly plant-based cells, for instance. They have like a more difficult membrane, which is the surrounding of the cell. So we would have to, use more harsh mesh methods, I guess, to really try to even break open the cell in the first place. Okay. So basically, what we would have here at the end of this is, let's say you have here like a glass of water, or some buffer, and then you have here all these cells. By the end of this, you should have here instead, like in the solution, a bunch of like tiny, 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 tiny pieces of this, cell that you basically got, right? And then after that, basically, before I go here into detail, the next step that we have is actually we would do centrifugation, okay? Centrifugation, if I could spell it, there we go. So what we would do here then is basically we would take this. So we would take this bottle with like all the different proteins and organelle and all of that, and we would put it in something called a centrifuge, which you can think of as kind of like a, you know, you can think of it like a carousel basically. Okay. So as an example, imagine you have here like a carousel. Yeah, let me just try to draw a carousel, but please bear with me. I'm not good with drawing, but imagine here's like a carousel, and instead of these chairs in the carousel, we have tubes. So these are our test tubes, all in the carousel. And what it does then is it basically spins super, super, super fast. And when it spins this fast, depending on the speed, what happens afterwards, so we have like something like this, and what would happen after that is basically all the different segmentations would fall to the bottom. Okay, so let's say we have a low speed. After the low speed, all the red dots are on the bottom. Let's say the red dots are the nuclei. Okay, so after low speed, all the nuclei are in the bottom, and all the green dots, let's say the green dots are the proteins, are still on the top. So what we can do then is, by the way, this is called the pellet. Okay, all the things that are then on the bottom, it's called the pellet. We then would basically take this out, so we would then basically delete it, I didn't want to do that, but I think you get it. So like I delete this part. I delete like this part. There we go. And after that, we put it in the centrifuge again. It spins super, super, super fast. And then all the green dots are actually on the bottom after that, okay? So these are then placed on the bottom, and this is our new pellet. So the pellet is now the green dots, all of these proteins. And this is how we basically separate them with the centrifuge, okay? The next thing that we actually do is we actually salt it out. There we go. So what we do there is basically we add like a salt solution into the water right here, or into our solution. And what happens when salting out is basically we have first, let's say water and protein, okay? And after adding salt to it, so we add here salt, there we go. And after adding this salt, let me just put it right here so that it's a bit neater. After adding the salt, basically what happens is we put it like back into the spinner, so we just like spin it again, right? And what happens is the proteins basically like clump together, okay? So imagine we have here this, I'll just take this for now. Imagine we have here like all these protein molecules swimming around. If we add salt to it and spin it, basically they would clump together like this. You see? So that would, of course, allow us for a lot easier extraction. But what happens after that is the next step is where we actually need to remove the salt again, remove salt. And to do that, there are three different methods. We can do dialysis, and we could do ultrafiltration, and lastly also gel filtration. Okay. And how I understood how these work, okay, with my very limited biological knowledge, I guess, is with dialysis, is you can think of it as like having a bag, okay? So imagine you have here this bag and the solution is inside. And in there we have like tiny, tiny, tiny pores, okay? And basically the methodology behind this is that the salt is tiny enough to go through the pores, but the proteins are not. So this is how we, like separate the salt again from the proteins. And for the ultrafiltration, it seems like it works similar, but it goes through a filter instead. So imagine you have here your solution, like drops, and then here's like a filter. And then after the filter, you have here a cup with salt. So here's the salt. And then hopefully on top here, you would have the protein. This is how I understood it. And gel filtration has like a gel column where basically what happens is, if you put your solution through, they're like these gel, thingies. And what happens is, okay, let's say red is salt. So if I have like the salt basically kind of not gets stuck, but it like it reduces its speed basically when going through the gel. But the proteins instead are actually able to pass the gel easily, very quickly. So these are the three different types of basically removing the salt. And this is a very simple way, I guess, to go from a full cell to basically having purified, or, to a specific degree, the proteins out of the cells themselves. Now, there are, however, two more small details that we could actually add to this. This might help us a lot. So actually, two things that we would add in the start before all of this is we would actually, so this would be basically before the centrifugation and all of that, we would add something called a pH buffer, right? And basically what this is it's like some type of molecule or chemicals that help the pH level, basically not shift that much. So if the pH is at 7, with this molecule, if something gets added or reduced or something like that, it basically kind of like stays at 7 instead of going, up and down. And also, we could also add detergents, and for the detergents, basically what would happen is it's like it's like kind of like a pill shaped thingy like this, where on the front would be like on the tail, what gets attracted would be like fat, so fat basically sticks then to the tail, and to the head would be then water, so this would be a way to separate the fats and the water too. A way to actually help with this whole thing is to add proteases or protease. I don't know how to say like protease like this, um, like inhibitors. And basically, what happens is that during basically cells have these things called protease, okay? And the problem with protease is that basically um, they destroy the proteins, okay? So protease chops up proteins, but inside the cell, while the cell is still living and all active and all of that, basically it doesn't really bother the proteins. But once it actually gets released, this then becomes a big problem. So we two ways to basically inhibit it is by adding cold exposure. So basically making it super cold and freezing it. So, or I don't know if it's frozen, but it's like very cold so that the proteins don't attack the proteins, because of course, if they're chopped up, then the whole pipeline is basically ruined, right? We want the proteins. And if we don't have the proteins, then why are we doing this? Or we can also use some chemicals called protease inhibitors, which, help in keeping them intact. Now, finally, I will just show, some quick methods on gentle, harsh, and cryo grinding, on how to do this. So this would be an example of how we were to do this with animal tissue, okay? So imagine you have here the animal tissue, you put it in this buffer water, right? And then you use here a potter. So potters, they look like this, where basically you then, twist it and like push it down, right? And this is how you would destroy the tissue into like small things, and then you can filter it. And then we continue with the workflow like we saw before. Another way to do this is actually through ultrasound. So we used such a device which breaks open the cells using ultrasound. We could also use something called a French press, where basically this is done with pressure, So here we have like the full, cells, and then it presses it through pressure down, and then you have here the product afterwards. Lastly, cryogrinding uses nitrogen and very cold uh, temperature to basically break open the cells. You can kind of think of it like blending frozen fruits, where basically there's a very big difference between, in putting in a blender, mushy fruits, that are warm, or frozen fruits. When it's frozen, it just breaks up, right? And then when it's mushy and all of that, it might be a bit more difficult, and it gets like more soupy and all of that. So yes, this has been an overview of protein purification, explained super simply, and thank you very much for listening, and I hope I was able to help.